invite everyone to take their seats so that we can get started with this second session and final session, regretfully, on this particular BioVision on biorobotics, making the best of man and machine. And what I'm hoping we'll do, we have two very distinguished speakers, Professor Erin Warwick, who I even mentioned in the last session, is the first cyborg in this world. He is a professor of cybernetics, not surprisingly, carries out research in artificial intelligence, control, robotics, and cyborgs, and he's published over 500 articles, but he's also truly has tested himself and put a chip in himself, so he is the world's first cyborg. And he will speak to us first, and I hope we can have him speak for 25 minutes. And then Professor Nicola Franceschini, who will speak after him, and he is truly European, from France, Grenoble, Max Planck Institute, etc. So you all have their CVs in front of you. And then after that, I would invite both Professor Warwick and Professor Franceschini to sit here, and our three previous speakers to join us. And we hope to have about 20-25 minutes of interaction between the panelists, given the enormous range and variety of what is being presented. So without further ado, please join me in welcoming back again, I'm happy to say, hopefully into his second home and second office, Erin Warwick and the cyborg experiments.
Thank you, thank you. Not got much time, so I'm going to be very quick. Just to say, I'm going to talk for a little bit of time, and then I'm going to let my wife come and say a few words, because she has been involved with some of the experiments, and it's thought this year people might like to hear what she has to say about them. You'll see how she fits in. So very, very quickly, cyborg experiments. Cyborg is this merger of human and machine, and I've just got 5 experiments for us to look at. First of all, class 2 implants. This is as much just to introduce myself to you. Class 2 is, there we go, we heard it working, radio frequency identification devices. This is going back to 1998, and here's a young version of me having an implant. This is from my local doctor, and what he's doing is putting this little thing, not the thing on the left-hand side, the thing on the right-hand side, implanting it into my left arm. And what this did for me was to identify me to the computer in my building. So as I walked towards the laboratory door, the door opened for me because the computer was tracking me with this implant. Coming down the corridor, the lights came on. Coming through the front door, it said, "Hello, Professor Warwick." So it was really good fun, whether it was a cyborg, but it's the sort of thing maybe you could do at home if you fancy a sort of implant. Now, something else that possibly you could do now, because this is what some of my students are doing. What I'm going to describe now, some of my students are also cyborgs in a way, and these are the projects that some of them are doing. So this is something you could try. I have 3 students now who have magnets implanted in their fingers. And you'll see the magnet being implanted here. Now, the interesting thing is, if you look at the guy who's doing it, you can see he's got tattoos on his arm. And that is because he is a tattoo artist. That's what he does. And this is problematic because we have to get ethical approval from the university to do this sort of thing. And the guy, the tattoo artist that does this, goes by the name of Dr. Evil. That's what he calls himself. And on the forms that we have to fill in for the university, it says who is carrying out the medical procedure. And we have to put Dr. Evil on the form, which the university authorities don't like at all. I mean, he is actually quite good at what he's doing. Now, here's one of the students, Jawesh. You can see the magnets. This is an X-ray of his fingers, so you can see the magnets in his fingers. And it will show you what we're doing now. On the baseball cap are ultrasonic sensors. Ultrasonics gives a good indication of distance. The output from those sensors is fed down to a coil of wire which is around the student's finger. So as the distance to an object changes, the magnet vibrates more or less dependent on how far the object is away. So the student can feel how far objects are away. So it's changing an ultrasonic sense, which humans are pretty pathetic. We don't have those sort of senses, and it's changing it into a touch sense. I have another student now who's doing the same with an infrared sense. Infrared is like heat from a distance. So what he can do is sense heat from a distance, which has immediate military applications. If you can imagine you're a soldier about to go into a room and you don't know whether there's anybody in the room, you can simply push your finger around and say, oh, there's somebody there and there's somebody there. You know exactly how hot they are and how many people are there and so on. This is Ashley. He won the Best Project Prize last year. What he's doing is communicating via his tongue. So this is something that could help people that have difficulty communicating, sending small current pulses into his tongue. And people are very, very quick at understanding this. We've tried it on all sorts of different people with ethical approval, I have to say, and people are very quick in learning to communicate, to understand signals in this particular way. Now for something very different. This is a robotics, bio-robotics session. Now I'm going to just introduce you to some of my robots. I have to say we had a microphone here for particular purposes and somebody's pinched it. But there we go, thanks very much. Now this is a clip from BBC TV called Inventions That Change the World. It's to introduce you to some of my robots.
One of the AI community's leading lights is Professor Erin Warwick.
Well, this is a robot head.
Dissatisfied with vacuum cleaners and robot dogs, Kevin's gone one stage further. His cyborg comes complete with
radar in its nose and an infrared top lip.
Is it intelligent?
Well, in a very limited way, I guess it is. It's sort of a basic instinct. What do I do with this information that I'm getting? How do I move? How do I operate if this signal is different?
So what's it doing now?
Well, if you clap your hands...
Oh, wait, over here. Oh, it did it.
It's making basic decisions. All we've got it doing here is programmed to make basic decisions based on that sensory information that's coming in.
Getting the information in is nothing. That's like picking up a book. That's not an intelligent thing to do. Reading the book and knowing what the words mean, that's surely the intelligence.
Many machines can read books nowadays, but it means something different electronically to the machine as it would mean to a human.
Though the cyborg head does a reasonable job of sensing, it can't really interact with or explore its surroundings. It's fastened to its robot arm. And according to Kevin, it's only as intelligent as a dishwasher. If that's a dishwasher...
Yes.
Where are these in terms of... Are they dolphins, chimps?
I think you'd have to say it's about the 50 to 100 brain cell area. So realistically, you're talking slugs and snails.
But a slug and a snail, they know to eat. They know. These things don't...
They have some innate abilities.
There's no self-preservation with that.
Well, we do have. The power supply, they don't have their own batteries. They each have power they're picking up from the floor here. That's where they're getting their energy. That's where they're getting their food
But
from.
if you turned off this section of the floor...
If we turn off this section, they have a basic instinct to go and look for some power somewhere else. They will keep going to do that until they find some.
So if I put him down...
It should go off looking.
So he'll have to go and look for power. And are you going to tell him to do that?
No, it's an innate response for that robot. It's a basic thing of his life. Yeah.
No, his lights have come back on.
Lights have come on.
So he's found power.
He's happy now that he's getting power. Away to go.
But these then are as intelligent as slugs.
It's different, but in terms of numbers of brain cells and that sort of thing, yeah, we have slugs. Probably you could say we're up to bees and wasps, the sort of tens of thousands of brain cells, is about as good as you can get. Yeah.
Bees and wasps.
Yeah.
Right.
Now, those robots you saw there have microprocessors for a brain. What we've done is replace the brain of the robot with a biological brain. So the right-hand side of the picture you'll see is the physical body of the robot. The left-hand side, it says MEA, multi-electrode array. This is a little dish in which has electrodes on the bottom of it, and we place neurons, brain cells, that first of all are taken from rat embryos. We separate the neurons using enzymes, and then we squidge them down into this little dish. The dish actually looks like this. The black rings are called Potter rings, the guy who invented them, and these are to stop the brain dehydrating. It is kept in an incubator at 37 degrees centigrade, and very, very quickly the neurons start sending out tentacles. They link together and become the dendrites and the axons effectively. So after about 7 to 10 days, we've got a two-dimensional brain that we're keeping in the incubator. We then link it to a robot body, so we give it its physical body. So what you're going to see now, this is a robot with ultrasonic sensors. The output from the sensor, when it gets near to an object, stimulates the brain. The brain will then think about things a bit and change the direction of the robot. So this is what it is supposed to do. Perfect. And this is what it's not supposed to do, is change its mind and turn around. It's only supposed to turn when it gets to a wall, and when it gets to a wall, it was supposed to turn then. But this is like a baby robot. This robot is just 10 days old, so some of the time it gets it right. Maybe it was a bit early, but it seemed to be all right. Here we go. Have a look here. Perfect. And then gets it wrong again. Now we can look at this. The brain is linked into the robot body for about 1 hour every day. And we can see how the brain changes, how the neural pathways strengthen over time. And if I now skip ahead, this is the same robot, the same body, the same brain, but after 2 months. So we're looking at the robot now after 2 months, and the brain has been developing. And what we can see is how the brain develops due to habitual learning, that purely the process of doing this same thing day after day after day strengthens the neural pathways that we can look at under the microscope, and you see it now functioning. So this robot has had a university education, and you see it performing now. I'm not sure whether it actually hits the wall at all. So the robot itself is a physical robot, and the brain of the robot is purely a biological brain. Now we also have human neurons to use instead of the rat neurons, and we are looking, rather than the 100,000 brain cells, which is typical in the two-dimensional array, in a three-dimensional structure, we're looking at something like 30 million neurons. So what we have here is a robot potentially with 30 million human neurons making its decision. The only brain, there's no computer that's changing the direction other than if you count the biological brain as being that entity. So it's a robot with a biological brain. 30 million, of course, is the number at the present time, but if we increase it and increase it and increase it, of course, there's no technical reason why we can't go up to the 100 billion and beyond, and give a robot a biological brain more powerful than a human brain. When we have a human brain, we look on the therapeutic side, more experimentation, and one of the areas where technology is used is deep brain stimulation, particularly for Parkinson's disease. I'm now just going to show a quick clip of the effect of deep brain stimulation. Typically, we see the electrodes are positioned right in the central area of the brain, And the result is as follows. So what you're going to see is somebody with Parkinson's disease and then the effects of electrodes positioned deep in the person's brain.
I'd like to show you in 2 minutes what was the status of Mr. Plessman when he was referred to us by his neurologist in Belgium. And you see him before surgery, without drug, unable to stand up by himself. He's helped by Patricia Limousin, who is a neurologist in our group, and he's not able to do anything of, as he will tell you, makes your life looking like normal. He's not able to walk. He has having dystonia, which are muscular contraction, deforming the different joints, and this is the best he could do without the drugs, with the help of people around him and mainly his wife and friends. He was referred because the drugs were not working anymore. He was not having any more improvement, and as he will tell you, he was just stuck to a wheelchair. This is after surgery. You see the tremor. We turn one of the 2 stimulators which were implanted in the electrode into his subthalamic nucleus, which is a tiny part of the brain, and you see that in a few seconds the stimulation induces a stop of the tremor. This is still visible on the other side. We will turn on the stimulator on the right side, and you will see something even more spectacular than that. You will see that the patient feels again free, and he resets himself in his chair in the position where he feels the most comfortable. He will obey Dr. Pollack in doing some tests, even if he feels that those tests are a little bit useless as compared to the feeling he has. He can do that almost normally. On the right side, should I do it longer, does he say? And we'll show it on the other side. And then he will walk. He stood up alone. He's without any drug. You see him walking, turning. It was a few days before the Vienna meeting on movement disorder.
Now this technology works well, as you see, but it does use electric power all the time, and the power packs last at the moment about 2 years, and then the patient has to go back into hospital to have the power packs removed, which is quite upsetting, and you can get infection getting in the wound. So what we're doing at the moment is working with surgeons at John Radcliffe Hospital, Oxford, to make the stimulator intelligent. So we're taking the electrical information from the center of the brain, the subthalamic nucleus, feeding that electrical information into an artificial intelligence system which learns to predict when the tremors are going to start before they start. And what we can get is a prediction about 20 seconds before the tremors actually start. So this is an artificial intelligence system that is effectively modeling part of the brain and then predicting what it is going to do 20 seconds before it does it, so that the stimulation can be applied only when it's needed, not all the time. And we reckon that we should be able to save maybe batteries so that they last 10 years instead of 2 years. That's the aim anyway. That's therapy, and in a way what perhaps is more interesting is, rather than just repairing problems, is to look at enhancing problems. And the problem here is that the human brain is pretty limited. Potential enhancements. Well, memory, obviously, we've been hearing in other sessions about that. Sensing, we are very limited in how we sense. Communication, of course, if you compare how you communicate, or I communicate with a computer, it's pretty pathetic, really. So the possibility of upgrading, of enhancing our communication skills, for me, is enormous. I was in a session today hearing about computers thinking in hundreds or thousands of dimensions, and the human brain being limited to 3 dimensions. Of course it is, and we have enormous problems with understanding things. Link your brain to a computer and potentially you can think in more dimensions. But it's difficult to decide between therapy and enhancement. This is Campbell Aird, who lost his arm due to cancer, but you see him flicking the switch in his left hand to control the robot hand, which is all a bit of a shame, really. What we would really like is for him to control the robot hand directly from his brain. And that's really my main line of research, is getting a bidirectional interface from the brain to wires and out to a robot hand. If we get a successful interface to take brain signals onto wires and pick up signals from a hand, great. We can replace an arm, we can replace a leg if the mechanics are okay. But more than that, we're talking of brain signals on wires, and once we've got brain signals on wires, we can clip onto those wires and send those brain signals wherever we want. So what it means is, if we can have a successful bidirectional interface, your brain and your body don't have to be in the same place. As long as you plug your brain into a network, your body can be wherever you want it to be. So there's the future you've got. What have I done about it? Here's a clip from BBC News.
Now prepare yourselves for this. A British scientist has become the world's first bionic man.
It's true. Cybernetics professor Kevin Warwick has had an implant attached to his nervous system. We'll have a look at it in a minute to discover exactly how the brain sends out signals controlling movement and even sensations such as pleasure and pain.
The secret operation involves surgically inserting an implant and cable into the left arm of Professor Kevin Warwick.
Well, the operation's been going about 10 minutes now. Spoken to Kevin, everything's going fine from his end, and the implant will shortly be about to go in.
The implant was fired onto a main nerve near the wrist. Wires from the implant were then threaded through a cable beneath the skin, exiting further up the arm to an electronic connector.
Effectively, what we've got is a link from my brain down the median nerve, through the wires and out to the connector. And then in the weeks ahead, we'll be putting on our radio transmitter receiver that we've developed at the university, and that'll be making the link by radio from my nervous system to the computer and back the other way.
This is what everyone hopes to see, nerve signals resulting from finger movement or even emotions. The medical aim is to help people who are paralysed, although some experts are unconvinced. But Professor Warwick is used to detractors, and experiments linking his nerves to a computer will begin soon. Sue Nelson, BBC News.
So what actually happened? Well, it was 2 hours of neurosurgery. I have to say, this was not, I didn't need this for medical purposes. This was a research project. I think every professor should do projects like this. What I actually had implanted was this. It's called the Utah array, also called the BrainGate System, and this was fired into the median nerves of my left arm. Here's sorry if you had a lot of lunch. This is my left arm during the operation. You can see 2 incisions. They push that tube down between the incisions, and then they pushed that array down the tube, removed the tube, hammered the array in my nervous system, and then for 3 months I had the implant in my nervous system with that little green connector, and the terminals on that were linked into my nervous system. So every time I closed my hand, neural signals we could pick up from the terminals were sent to the computer to operate pieces of technology, but also we could send signals into my nervous system that I learnt to recognize. So I've got a little clip from Discovery to show some of the things I could do.
One man in tune with technology is the world's first cyborg. Part man, part machine. Kevin Warwick, Cybernetics professor at Reading University, England, took a leap into the future as long ago as 2002. For 3 months, a silicon chip was implanted into the nerves in his arm.
At that time, no human had had an implant of this type before.
It was a procedure that until then had only been tested on chickens.
Would the body reject it? Would it affect the way I moved or my sensory capabilities? I could have lost the use of my hand. Could it affect my brain? Ultimately, could have sent me crazy.
Kevin was plugged into a computer which monitored the nerve signals from his brain to his arm, receiving and transmitting them as radio waves. With the signals from his brain, Kevin could not only turn on lights, he could control a wheelchair. And from 5,000 kilometres away via the internet, he succeeded in getting a robot hand to mimic his own hand movements. His most impressive experiment involved his wife, Irena, also having a chip implanted in her arm.
The communication experiment between myself and Irena was vitally important to me. This was the highlight of the whole experiment.
Kevin wanted to discover if it was possible for his brain to receive and feel signals from Irena's brain.
When Irena first moved her hand, it actually felt like a charge running up my index finger. It was a phenomenal feeling. Literally, every time she moved, my brain received a signal. There we were communicating for the first time in the world, nervous system to nervous system.
It was the first time two people had electronically communicated by thought alone.
In the short term, we're going to see implants used to help people who are paralysed switch on lights, operate the TV, just by thinking about it.
In 30 years, he believes we'll all have chips in our brains.
But ultimately, having extra memory, having downloads from the computer, uploading from the brain.
So, just to conclude with a few holiday pictures, and then I'm going to introduce my wife. Ultrasonic sensors, extrasensory input. This was fed into my nervous system, so with a blindfold on, I was able to detect how far objects were away, and I'd move around and not bump into them. My wife helped in a number of ways. Here she's wearing some jewellery a student at the Royal College of Art in London designed. It changes colour, the jewellery changes colour from red to blue, and it was linked to my nervous system. So if you can picture the scene, when I'm calm and relaxed, my wife's jewellery is blue, and when I get excited, I can see you get where this is going. When I get excited, my wife's jewellery starts flashing red. So she doesn't work in the university. There she is, jewellery's blue, fine, he's not doing anything he shouldn't. And then the jewellery starts flashing red. What is he doing? And more importantly, who is he doing it with? It's not true. I'm an innocent person. This was taken at Columbia University, New York, where the guys there helped me plug my nervous system into the internet and link to the robot hand in UK. So I moved my hand in New York, my neural signals went across the internet to move the robot hand in England. When the robot hand gripped an object, signals were sent back from the fingertips of the hand across the internet to stimulate my nervous system in New York. So essentially I could feel how much force the robot hand was applying on another continent. And then the most important thing of all for me was when my wife got involved with the project as well. She had, as she'll describe, electrodes implanted so that we could communicate nervous system to nervous system. And now I'm going to introduce my wife. Yes.
Hello, I'm Irena, Kevin's wife. I was asked to express my feelings about all his experiments. I'm not a scientist, and this is my very first presentation, so I'm quite nervous. How it started, it was when he planned to have his first implant, and I realized I didn't have any clue about all implants, and he explained to me, and I started to worry about him because it could be infection. And one example was when the doctor wanted to sterilize the implant, he put the implant to the boiling water and it burst into pieces. So you can imagine what everything could happen. But after that, he was planning the big serious implant to link his nervous system into the computer. And more we talked about it, I wanted to be part of it. Once I was skeptical, I wanted to see on my own body if it was working, like he was saying, how were discussing about feeling our emotions and phobias. And mostly, I wanted to wave flag for women. And so what I had were 2 wires pushed into my median nerves, into my left arm. And you imagine that I felt it would be very painful. So I asked the doctor if I would have anesthetic. And he said, no, we have to make sure that we have the good contact there. So he pushed the first wire there, and I must admit I screamed. And he pushed another one, and then we went to the laboratory, connected each other to the computer, to different computer, and it was exciting because when Kevin moved the hand, I felt pulse in my palm, and it was sort of like slight lightening. So every time he moved the hand, I felt the pulse in my palm. So it was communicating first time, nervous system to nervous system. And next implant he wants to have into brain. And I must admit I'm scared. I don't want to be part of this. So he will have to find somebody, some other person, but I would prefer if it wasn't a woman because I would be jealous. Thank you.
Wow. Thank you. But I kind of agree with Mrs. Warwick. There has to be still a question of something called privacy. I mean, if you connect brain to brain, all the private thoughts that you could possibly have would be, well, I don't know. Kind of opens strange possibilities. But at any rate, with that, let me move on because we have limited time, but I would like very, very much to invite our speaker, Dr. Nicola Franceschini, who will speak from animals to robots and back again. So, Professor Franceschini, microphone's yours.
So first of all, I would like to thank Ismail Serageldin very much for this invitation. I'm so honored and so happy to be here in this famous city where so many prominent scholars have been living and worked in the past. So we are working in the Biorobotics laboratory in Marseille, and I want to bring you a small story from our laboratory. There are several stories, but I selected this one just to show that by looking at animals, one may get really some nice inspiration to build robots. And the main point for us, because we are very interested in biology, the main point for us is that it raises new biological questions. And I will come back to that at the end of my talk. Now, I would like to introduce something which is, in fact, quite common sense for animal navigation. Maybe you are not aware of that, but not only insects, but also humans and animals use what is called the optic flow to navigate. You know very well some examples of the optic flow. That is what you see from the window of the train, for example, and you notice something, in fact, quite important. Namely, those objects that are far away, the objects that are far away, they slip on your retina at a very low angular speed, while the objects quite in the front here are slipping at a very high angular speed. So this is what is called the optic flow, and in that particular case, the translational optic flow. An optic flow is, in fact, the angular speed omega in radian per second, or you can also say in degree per second, at which contrasting features move past the eye when you move, when you locomote in the air or on the ground. The optic flow laws have been well established over the past 50 years, and in this particular case of the translational optic flow, you see that if you are, for example, an observer going in this direction, this is your heading direction, and you see that everything on your left-hand side or on your right-hand side, depending on the angle phi of your heading with respect to these objects, and depending also on the distance to these various objects, they move on your retina, they move at a particular angular speed. And all these vectors here reproduce in length and in direction those contrasts that are moving on your retina. The important point here is that we humans and animals in general, we need motion-sensitive neurons in order to be sensitive to these. We need motion-sensitive neurons, and I want to show you that if motion-sensitive neurons have been discovered in animals like mammals and also in insects over the last 40 to 50 years, we can now start understanding the underlying circuits, so to speak, and we can go then from neurobiology to electronics, and from these electronic optic flow sensors, we can go up to robots. Now, the animal we are dealing with is not a human, not a mice, not a cat or a monkey. This is something, is a more humble creature. But these creatures are just fantastic. They have a mass of less than 1 gram, a size of less than 2 centimeters, a span. The ground speed may be up to 10 meters per second, much faster than humans, and rotational speed up to 4,000 degrees per second. Basically, in each eye, in each compound eye, you have basically 3,000 facets, for example, in the case of the fly, which means 3,000 omatidia. These tiny eyes, we call them omatidia, 1 omatidium, 2 omatidia, which means also they have only 3,000 pixels in their eye, which is very low with respect to a common photographic camera that you can buy now for $100. And they have, in addition, they don't have 100 billion neurons in their head. They have only 1 million. I say only, but 1 million neuron is a lot. And most of these neurons are actually devoted to vision. And these creatures can do obstacle avoidance, smooth pursuit, hoverflight, accurate landing, just great feat in behavior. And if you look inside the head, obviously, it is not easy. It is very complex. And we are far from understanding everything of this brain. We start understanding the omatidia here, which you see elongated here. We start understanding the lamina, the first optic ganglion. We know very less about the medulla, and we know a lot about this third optic ganglion, the medulla, where precisely our optic flow sensors are located. If you imagine this experiment when you push a microelectrode through a tiny hole that you may make in the head capsule at the back, you will successively encounter approximately 60 neurons. All of them are sensitive to motion. And from these 60 neurons, I'm showing you 23 of them from this work here. 23 of them shown in exploded view along the median axis of the head. You see that if you would push the microelectrode, you would first encounter some neurons that are sensitive to vertical motion in the direction of the black arrow here. If you push the electrode further, you encounter these 10 neurons which are also sensitive to vertical motion. And recently, my colleague in Munich, in Boss laboratory, showed that you can stain some of them. For example, from these 10, 3 have been stained with a microelectrode containing green fluorescing dye, and at the same time, you can stain the postsynaptic neuron that will gather the information from here and bring it through the neck, where? To the head and to the muscles to control the legs and the wings. So we have to do with a very complex machine, and we are just starting to understand, in particular, how motion detection can be made, can be organized in the underlying neural circuits. Now with my colleagues Alexandrina, a long time ago in the 80s, we recorded a lot from this H1 cell, which, as you see, is sensitive to motion in the direction of the arrow, which means from the back to the front on the head of the animal. And if you look at the response, you see something quite remarkable. You see that as long as you show motion in a particular direction, from the back to the head, the arrow here, you see that these neurons, which are in fact spiking neurons that generate nerve impulses, like so many neurons in brains, and here is plotted the frequency in impulses per second, the frequency, you see that as long as motion takes place, the frequency is relatively high, maybe 130 hertz, 130 impulses per second. If you stop the motion, immediately the response goes down. And if you move this pattern in the wrong direction to the right here, you see that the response is nearly inhibited. Now, there seems to be a machine underneath. And indeed, it's a beautiful machine because you can record this recording here during days, during several days. Of course, you have to feed your fly under the microscope every evening, but then in the next day you come back and you continue recording the same kind of stuff. Now, the problem with these big neurons, as we have seen, are that they collect inputs from thousands of so-called elementary motion detectors. And we were very interested in those days in understanding how one elementary motion detector, one of the thousands elementary motion detector feeding these big neurons like H1 would work. And to do that, we were very much helped by a discovery that we made in the 80s, namely that we can access the retina of the fly in vivo. What you see here is the beautiful autofluorescence colors from the photoreceptors. You see, this is the content of each facet. Behind each facet, this means within each ommatidium, you have this small group of, say, 7 or 8 photoreceptor cells. Some of them are red fluorescing, some of them are blue or green fluorescing, some do not fluoresce at all, some central cell even fluoresce red. I don't want to go into the detail of that, although we have analyzed that during days, or I should say perhaps decades, but we know that these central cells here, number 7, we call it number 7 here, and its prolongation, which you don't see here, number 8, are done for color vision. Whereas these red cells here are done in particular for motion detection. You see how there's a beautiful, distribution of labor among the retina with color vision on one side and motion vision made by other types of cell. And the idea to analyze motion detection was simply due to this. These cells here are in the back focal plane of the facet lens. They are here, back focal plane of the facet lens, and each of these cells, only 3 are represented in this longitudinal section, each of them is looking in a direction, different direction in space. They have divergent optical axis. And think of what vision is actually. When this contrast, my hand, is moving in front of my eye, try to imagine what occurs in the depth of my eye on my retina. I move like this, and the image is moving like that in the back focal plane due to the inversion of the optics, obviously. Now, what we did is to simulate a micromotion by applying a 1 micron spot onto this photoreceptor cell, and later on, a 1 micron spot to the other photoreceptor cells. In other words, by using a sequential stimulation of 2 neighboring photoreceptor cells, we should be able to analyze and to record the elementary response to motion. That's what we wanted. And to do that, of course, it's not quite easy, and we had to build a special instrument, which I call the microscope telescope. You see the fly here very carefully in the center of this big instrument, which can turn in azimuth and elevation around the animal. The microelectrode is presently recording from the H1 neuron here with the amplifier and so on and so on. The spikes that we record from that are very small, 200 microvolt. You have to amplify considerably in order to get a signal. And indeed, the microscopic part allows you to select a given facet. This one, for example, you see, we are not far from the frontal border of the eye, but you could select any facet. And within that facet, remember, there are these 7 photoreceptor cells, of which we are going to stimulate only 2 with 1 micron spot, because they have approximately 1 micron in diameter. And the result is this: when we stimulate these 2 photoreceptors sequentially, as shown here, you see, number 1 first, and then number 6, you get a beautiful discharge of impulses, of nerve impulses. And whenever you reproduce the sequential stimulation, you get a nice burst of spikes. Now, if you reverse the stimulation, starting with the cell number 6 and ending with the cell number 1, this is what you see here, you get no response. You can imagine that we are here close to the elementary principle of motion detection. Just want to show you one other response. Here, it's not the spikes themselves, but again, this is the frequency of the spikes that is plotted. And you see, if you stimulate 1 and then 6, you get a beautiful response up to 50 or 60 hertz. And if you stimulate in the other direction, you see that the response is even inhibited. I don't want to go into the detail again of that because it took years in order to try to figure out a model of this motion detection circuit. I just want to say that very rapidly in the mid-80s we had reproduced, we were inspired by this to build our first electronic motion detectors. You see here 2 photodiodes, for example, behind a lens, in the focal plane of a lens, maybe a 5mm lens, for example. And you see they have a receptive field which is more or less Gaussian. So if you imagine a contrast like with the border of my hand, that would be moving here from the bottom to the top, they will obviously successively excite the photoreceptor number 1, and then later on, after a time delta t, they will excite photoreceptor number 2. And you see the result of the signals from the photoreceptors will be separated by delta t. So we have developed ways of analyzing this principle so that in the end, finally, the sensor gives a signal that is proportional to delta phi, the angle here, divided by delta t, which means the sensor gives the optic flow, omega, as now this circuit, we have devised it over the years in various technologies. This was more or less classical surface-mounted technology, and all of them are at the same scale here. Here you see this is rather made with Co-Fi Ceramics in collaboration with the University of Oulu in the framework of a European project. And this was made here more recently with 25 pixels. This is a purely analog VLSI retina, which was done in collaboration with the Center for Particle Physics in Marseille. Now, early on, very early, in the mid-80s, as we had these first motion detectors, we endeavored to incorporate that onto our first robot. And this was a big challenge. And this is the head of this robot of those days, in '89. And you see that there are many facets, approximately the same number as an array of facets in the eye of the fly. And behind each pair of facets, you have one such card, like this one, a conventional analog card. So, and they measure the relative motion that occurs between the head of the robot and the environment. And so the first robot that we built is this one, the RoboMouche, the RoboFly, so to speak. And basically, by moving forwards, the RoboFly creates a translational optic flow to which the compound eye is sensitive by means of this array of elementary motion detectors located here. And secondly, from the measured optic flow, the robot determines the distance to the obstacles and can therefore avoid them and drive around them at a relatively high speed, which was in those days already 50 centimeters per second, half a meter per second. Now, this was good, and perhaps we can see very rapidly some behavior of this robot. See that it's practically a horizontal slice across the eye of the fly. And here the robot goes to the target lamp here by avoiding all these pores, which are purely randomly arranged in the laboratory. So it sees them only on the basis of the relative motion between itself and the surroundings. In other words, it sees them on the basis of the translational optic flow. Now, this was a successful project indeed, but I must say that in a way this robot was cheating, because we had an optical encoder on the wheels that was able to give the robot the value of its speed. The big problem is, what is it when you have an aerial creature that has no contact with the ground? Terrestrial creatures have contact with the ground. We have contact with the ground. and by means of planetary receptors in our feet, we have, in a way, an access, possibly, to our own speed. But this is not the case for aerial creature. And I just want to make a remark about aerial creature like aircraft and so. And you know that to prevent crashing, conventional aircraft need to measure their ground speed, for example. They do with a radio altimeter. To measure their ground speed, ground height, or radio altimeter, ground speed with GPS or Doppler radar, and to measure their descent speed with a variometer. Now, do insects and birds really need such sensors? And if not, how do they estimate their ground height and their ground speed in order not to crash at all times? Now, what I want to show you here in a few minutes is that the principle we have introduced, which we call the optic flow regulator, allows the insect to navigate without knowing its height and without knowing its speed. The source of inspiration is actually very old. It dates back to Kennedy's hypothesis. In 1951, Kennedy said, not John Fitzgerald Kennedy, of course, another one. In 1951, he said, migrating locusts maintain a preferred retinal velocity with the ground below. So a preferred retinal velocity, we would say today, a preferred optic flow. So try to imagine the problem that an insect has to solve when flying over a terrain like this one. You have all these contrasts that are sweeping under you, relatively sweeping. Look at that. If we can see it. This is a really major problem, that these insects have to evaluate the optic flow that they get from the bottom of their eye, from the ventral ommatidia, so to speak, when they fly at a relatively high speed, as I said before, up to 10 meters per second. But let's try to formalize this system. You see it here, an insect which is flying at a given height, 1 meter or 2 meters, for example, and at a given speed. And you discover that the optic flow is nothing else than the ratio between Vx and h. Now, what you heard before is that flies and insects in general, and probably birds also, they have these so-called elementary motion detectors, the EMD, and here one EMD is represented behind a single facet, as we analyzed them before in the fly. And you see that at the output of the EMD, you have an omega measured signal. So the animal actually can recover the ratio Vx over h. Of course, by measuring this signal here, the insect does not know its speed, does not know its height, but at least it knows the ratio between the two. And the Octave autopilot that we have proposed, and Octave means optic flow-based control system for aerial vehicles, is this one. Octave, this autopilot principle, attempts to regulate the optic flow measured, that is, to maintain the optic flow constant and equal to a set point omega set. Secondly, Octave does so by controlling the lift. Octave is a feedback control system that we call the optic flow regulator. The sensory feedback is provided by an optic flow sensor, like the EMD that we analyzed in the fly. So let me describe you this feedback system. Octave is a ventral optic flow regulator. It does not regulate the altitude. It does not regulate the speed. It could not do that. There's no servoing of the altitude, no servoing of the ground speed, in contrast with conventional aircraft. Look at what occurs when you have an insect flying here. On the right-hand side, you see that the insect basically produces with its beating wings a mean flight force. And this mean flight force can be decomposed into a tiny forward thrust and on the vertical axis, a vertical lift that will sustain the animal and also allows it to climb, to raise in the air. Now, this same angle that you see, the angle theta, you see it again here in this so-called block diagram. And when the pitch angle is a few degrees forwards, nose down, then it gives rise to a tiny thrust that, via the surge dynamics, will give rise to a given speed. Likewise, the vertical lift via the heave dynamics will give rise to a flight altitude. Now, we have these two blocks. One of them will control the speed, ground speed, and the other one, the lift, will control h, the altitude. And what we think is that simply on board the insects, the ventral optic flow is measured by means of these elementary motion detector as optic flow sensor, and it is compared at all times to a set point of optic flow. So there's a resulting error signal that finally will control the altitude h, the ground height. This is how it operates. And you see this is a typical and a very simple feedback loop that will control h at all times. So we immediately set up this control system on board a very elementary helicopter that we tinkered in the laboratory. So we modified the rotor speed to get more lift if necessary. But basically what this helicopter has is an eye. You see the eye here, a single facet if you like. Behind this facet there are only two photoreceptors and a single elementary motion detector. We don't have thousands of them, but this is just to illustrate a principle. Now, if you launch this robot here, you see that during this phase, the robot was slightly pushed down, was slightly tilted forwards, so that it gained speed. And because it gained speed, it had to go in the air. It had to raise in the air due to the feedback. Now, at some times, obviously, due to the drag, it will reach a level flight. And you see that in the last phase, between these two vertical arrows, what we did is that we pitched the robot backwards so that in the end, it would be really vertically oriented. If it is then vertically oriented again here, there is no forward thrust. There is no acceleration forward anymore. And the robot will land. It will land gracefully. We show that it lands gracefully. And we can even show that it must, and it does, land at a constant speed. You see, this is the terrain over which, a random terrain over which this robot, this real actual robot, was flying. Now, let's have a look to the perturbation. There are several kinds of perturbation to this feedback. There's in particular the wind speed. For example, if you face a headwind or if you have a tailwind, this can be rather embarrassing. This is a big perturbation. Now you have the relief altitude. For example, if the robot is to fly over a soil that is slightly, gradually climbing, a kind of ramp, if you like, then you see what will occur, because gradually the ground height will be lower and lower when you fly over the ramp, so that omega is too high with respect to the set point. So the error signal is large and big and positive, and with it will raise the altitude so that you will automatically get terrain following. And this is what we see here in this video. You see Frank Ruffier here in front of the Octave robot. You see that we installed a kind of ramp in the laboratory, and the robot climbs over the ramp, and immediately after it comes down again. The only thing it has as a sensor is the optic flow sensor. It has nothing else than optic flow sensor. It does not know its height over the ground. It does not know its speed over the ground, but it behaves conveniently. So, just a general conclusion about these various behaviors. We have just seen the terrain following over a ramp like this one. You see that all these trajectories are very well superimposed. You can do this experiment many, many times. It's always the same, and we never had any crash in the laboratory, showing that this is a rather robust method. Evaluating the optic flow is a very robust method for avoiding the ground in this particular case. We saw also the automatic takeoff, and we saw the automatic landing, which I don't have time to explain why, but it must land at a constant slope, and it does land at a constant slope. Finally, a very nice reaction due to the perturbation of a headwind. You remember that you have to subtract the headwind from the airspeed of the robot. And this, of course, this means that the speed of the robot will be lower. And because the speed of the robot is lower, it has to go down. And you see here, it does go down at a very small altitude. And if the wind is strong, what occurs is that it is so strong that the robot gets such a brake that its speed gets very low, and so the altitude must get very low, and this is what we get, a kind of forced landing. But it is again a safe landing because it occurs at a zero vertical velocity and zero horizontal velocity. Back to insects. How can this? You are probably doubtful that it could ever explain something about insects. But it is all on the contrary, because it explains in a way the terrain-following abilities that was demonstrated over the years. It explains how honeybees land at a constant descent angle, as was shown about 15 years ago. It explains that insects descend in a headwind and ascent in a tailwind. And finally, it explains the very strange observation of Heran and Lindauer in '63, and they showed that when bees were coming back to the hive and they were encountering a lake with very calm water, they went down and they drowned into the water and never came back into the hive. So in conclusion, this explicit control scheme, this feedback system that we proposed, accounts for various insect behavioral patterns observed over the last 60 years. Octave is therefore an interesting working hypothesis, as it may explain how an insect can fly without any needs for measuring its ground speed and any needs to measure its ground height. And this is a very important point that necessarily can give rise to potential application. Because if there is no need for any explicit knowledge of ground speed, you don't need a radar or a GPS. If there's no need for explicit knowledge of ground height, you don't need a radio altimeter. If there's no need for an explicit knowledge of descent speed, you don't need a variometer. This means that Octave autopilot saves major avionic sensors, which are reputedly heavy, costly, and power-hungry, because they are active emissive sensors, usually, on board aircraft. So this is not only consistent with the insect's limited avionic payload, but it may offer interesting aerospace application. And one of them I just want to show you. In recent time in our laboratory, we had a simulation in collaboration with the European Space Agency of a automatic landing on the moon. You see a moon lander here. The moon lander has retro rockets, and the retro rockets obviously spit in a given direction so as to brake the lander. But at the same time, by means of some inertial system on board, we gave the lander the command to pitch at 0.275 degrees per second. And so this means that the lander is raising its body more and more. And due to that, you see that the horizontal speed decreases, and so does the vertical speed, Vz. Now, you see the principle is the same. What is essential is that the ratio between the two remains constant. The ratio between the speed and the height remains constant. This is the optic flow, and in that particular case, it remained constant at 0.3 radian per second. Now, let me finish by concluding that you have seen one example of this biological inspiration that may lead to biologically inspired sensors, autopilots, and even robots. And as I said at the beginning, we are very much interested in the fact that this raises new questions and opens new hypotheses and obviously generates new behavioral experiments. Now, as a general conclusion, I would like to say that implementation of a biologically inspired principle on board a mobile robot allows us to test its validity and to appreciate its robustness or its fragility, I should say. This reconstruction approach is much in the spirit of what Richard Feynman left on the blackboard of his office at Caltech when he died in 1988. The students found on his blackboard, "What I cannot create, I do not understand." Try to meditate about this. And finally, most important point, biorobotics creates a salutary synergy between scientific disciplines that used to talk little to one another: physiology, ethology, ecology, optics, mechanics, informatics, electronics, control theory. And this multifaceted science prevents narrow thinking and fosters rational and critical thinking. So this is much in the spirit of the early scholars in Alexandria, and before all, it is ideally suited to the education of our young students. Thank you very much.
I would urge, please, Kevin Warwick and Professor Franschini take their seats, and I invite Professors Arkin and Sinibaldi and Professor Nakamura to please also take their seats. We have a few moments for interaction with our colleagues. I've collected some questions, but I also, of course, we have some that have already been given for them. So with that, yeah. Okay, really, I must start that we've had now 5 presentations on cutting-edge science in many, many ways. And 2 inherent questions are there, and they were touched upon by some of the presenters, but one of those questions has been at the heart of Professor Arkin's presentation, which is the ethics of the experiments. And I note that Kevin Warwick several times said that they obtained permission from the ethics part of the university for the experiments. But a common theme from many of the questions that came to me was about risks. When you're doing cutting-edge research, you're taking risks. In the case of Kevin Warwick, he took his own risks rather than made it out on others. But I can ask Professor Arkin, the run amok part, robots running amok is a sci-fi. Is that a real risk or not? One of the questions came to me about manipulating humans mentally, not just physically, through distance connections. That one is really more for you. And so the risks and the ethics of taking risks in experimentation. Professor Arkin, would you go first, and then we'll go to Kevin, and then we'll come back to our other colleagues.
There are many concerns with the deployment of these systems. I put a few up there. It was interesting you picked up on the sci-fi one as well. Hollywood has prepped us for that particular response through the Terminator movies and almost every, at least Western movie. The Japanese are more benign, I would think, in their treatment of robotic systems. Has a robot that goes amok. Starts killing people, whether it's RoboCop or Terminator or Stealth or what have you. In some cases, it actually comes back and is good at the end. Is there a risk? Of course there's a risk. There are human soldiers, unfortunately, as you know, in recent times that have run amok as well, too, let alone whole squads, troops, in some cases even armies that risk whenever there is an armed conflict persists. The question is the risk manageable? Can it be less than with human warfighters? At this point in time, I believe it is. Part of it has to do with the architectural design that I referred to, something referred to as an ethical governor, which I didn't have time to talk about, which actually makes the decision as to whether a particular platform has permission to fire or not. Can that potentially be reprogrammed? Yes. Will that be documented in evidence for a potential war crimes tribunal? Conceivably so. Probably much more inspectable than for a human's brain in terms of whether an intention to violate a particular international humanitarian law edict existed. So certainly there is a potential. I don't believe, at least in most existing views of the military, that hordes of robots sweeping across the countryside are the vision of the future. There is something referred to as the warfighter ethos, which argues that a human presence must be maintained within the battlefield. And for this and all these reasons, I am dismissive, as you stated, regarding science fiction.
Okay, Kevin, how about you?
Yeah, I mean, there are risks associated with experimenting on yourself. It's a very different thing. Things could go wrong and you're injured. In terms of risks with robotics, where you're putting intelligence into robots, typically it is more of a computer type of intelligence. And there are big differences between computer type of intelligence and human or animal intelligence. The sensory input we've been hearing about several times today, multi-dimensional thinking and so on. So the thought that this intelligence you're putting in a robot is going to behave in the worst case like a human is really being extremely naive. And I think for any artificial intelligence expert to try and pull the wool over your eyes and say, oh, it's perfectly safe, it's a risk that you might believe them. That's the biggest risk. The point is, this is a different type of intelligence. And as Ron mentioned, the Terminator, I mean, in there, it was said you can't bargain with it, you can't reason with it. The robot with its own computer type of intelligence is thinking in its own way. It has its own values, it has its own priorities, and you as a human are probably not one of those priorities. So I think there are big risks in putting intelligence in robots that have a military capability. And allowing them to adapt and learn and make their own decisions is a big risk.
Well, in that case, let me just take it a step forward, further and say, I think we all remember the Asilomar conference back in 1972 when recombinant DNA research was scaring everybody. And the scientists got together and put some rules. And I remember at the time I was just finishing my thesis in Cambridge, Massachusetts, and the city of Cambridge was very concerned, and people were scaring them about Frankenstein monsters coming out of the labs at Harvard and MIT. And then when the scientists put together their standards and the P1, P2, P3, P4 levels of protection for different kinds of experiment, the city just adopted that ordinance and the universities went along with it. So, you know, the E. coli that die is outside of the Petri dish automatically and so on. Is there in robotics and biorobotics similar built-in safeguards that we can ethically ask that all experimenters taking risk right now should observe? I mean, pretty much as we did with transgenic and biological research. Now, I emphasize ethical researchers because obviously somebody, I mean, the fact that something is illegal or unacceptable doesn't mean somebody is not going to do it, but at least that generally speaking, funded research, published research, etc., should address that. Is there such a thing? Anybody in the panel?
I'd like to speak to that because the Asilomar workshop was a watershed event in
Yes.
bioethics.
It was, yes.
What's interesting about that, though, compared to what's happening in autonomy, is there was a marked discontinuity in capability with the advent of recombinant DNA research. People stood up and said, whoa, we can do something could happen that's fundamentally different right now. The progression of autonomy in these systems is much more insidious. We're gaining capabilities slowly over time. Where I don't expect the invention of a positronic brain, for example, which will suddenly have this, to invoke science fiction again, suddenly a leap of capability. There was a workshop in Berlin not too long ago, held by the International Committee of Robot Arms Control. I was there at a meeting where they actually called for an outright banning on these particular types of systems. And I encouraged them to think of a moratorium because I thought that might be better accepted by more people at this point in time, which is exactly what they did in Asilomar. They called for a moratorium until they could come up with the right set of laws and guidelines to be able to do that. I think that's an important step. The answer is, are there guidelines? There are general ethical guidelines by the professional agencies, but they are not specific at this point in time and not specific enough.
Well, it seems that the diversity of what is going on, from Professor Nakamura to Professor Franceschini to Professor Sandini, that from I don't know, a robotic octopus would probably be able to escape from almost any environment if it has intelligence and doesn't require to live inside water, or even if it did, it might go into your drain system or whatever. As far as I can make out, I mean, there's neither a moratorium nor actually existing rules. It's really up to a case-by-case basis in various institutes. Is it so in Japan, or is it different?
Well, I think the ESCO issues is mixed here. I think as far as in Japan, the ESCO issue is very conservative, but there are several different ESCO issues discussed today. I think ESCO issue is the battlefield or weapons, is more asking about the ethics of humans. What is acceptable for humans? So it's human ethics. And the another one would be if the scientist can predict the high danger for new technology or new science. That is the risk issue, if it is acceptable or not. I think the ESCO issue of humanity and the ESCO issue of risk is different. Also, for scientific community, the accepting the new experiment is again the different ESCO issue. It may be acceptable as long as a person likes, but in the scientific community, we should ask if we want to see those kind of experiment repeatedly, and if that is creating new knowledge.
Okay. Yes, go ahead. Yes.
I may add. Yes. So I think that the point you raise is really a reasonable one, and the quest for a safeguard is quite widespread. And for instance, I think there is no general answer to that, no general approach. For instance, as for the medical application field, you mentioned the octopus, and in that case there is the softness which could be used as a safeguard against injuries with the operators in the clinical room. However, as Professor Warwick mentioned, there are many types of intelligence. For instance, you can use also rigid robots, classical robots, and endow that robot with many cognitive layers. And this is something drawing the attention of the community now. So there is to make these systems more intelligent, to understand which is the surgeons operating in the room, which kind of gesture skills it has. So endowing the system with more cognitive layers to raise the level of safeguard and to manage better risks. This could be an issue which could be applicable in more fields.
Well, Professor Franceschini, does CNRS enforce any rules?
Well, no, I'm more fascinated by the fact that throughout the world now humanoid robots are really in, and everybody try to work on humanoid robot. I'm not quite sure that for the use they might have. For example, if you think of if you are ill in some clinic, some hospital, and if you see a humanoid maid that would bring you your medicine, I'm not sure it would be better it would be better to have a humanoid robot rather than an octopus robot or a dust cleaner robot or something like that, because you would accept it as something different. But if you see really something like nearly looks like a robot, I think it's rather frightening.
Well, I'll come back right now. We'll have two other questions for all of you, but just for the audience, I remember being actually in Lyon in BioVision. Sometime I was chairing a session on biorobotics, and this was still a few years ago, and they showed this image of a robot doing a brain surgery. And somebody said, how can you possibly entrust the life of a human being to a machine where it might go wrong? And my answer was, well, you all fly all the time, And, foggy weather is very common in Europe. And today, almost all plane landings are handled in foggy weather by robots. And in fact, the Boeing allows a human override if twice expressed, and the Airbus doesn't. So every time that the plane lands, 300 human lives are entrusted to a robot. So why are you nervous that we entrust one human life to a robot? And there was total silence, because that's Ron's point about insidious growth. It's not a sudden leap. People hadn't realized that from automatic pilots, we had gone to robotically controlled or computer-controlled landings, and this was everyday affairs. And today, of course, we showed cars without drivers and all of those things. So the reason I raised the risks and the ethics is because we're all witnessing enormous new power in terms of the new biological techniques and scientific breakthroughs, enormous new power in computing and computing capabilities, and enormous new powers in nanotechnology, and all of that is converging. It's opening up all sorts of new fields, and the fact that it's not totally discontinuous doesn't mean that at a very accelerated pace, we don't need to say, oh, wait a minute, we need some new thinking about this. And that's where I come to the new thinking. Question to Kevin, which came from the floor. We saw the medical application to the Parkinson's disease, but is there a direct medical application from communication between, or that you can foresee, between nervous system to nervous system?
Yeah, I mean, it's one of those things we don't know where it's going to go. The possibility of you and I connecting our brains together and start communicating like that is a realistic one. What that will mean for society, the dangers that it holds, the commercial opportunities that it opens up, we don't know at this time. So to say, no, you shouldn't do that experiment, there's not really any negative things to say about it at the moment until we do the experiment. I think there are other possibilities. I mean, let us say if I took Ron's brain and put it in a robot body, a nice shiny new body. I mean, you might like this. Now, if all this robot is going to do is to write academic papers, probably nobody, well, you know, whether anybody would read them or not, I don't know, but probably nobody's bothered. But if then we gave Ron, you know, some armory, some weapon systems, some bombs, and he has a drink one night in his robot form of oil, probably, and then starts blowing people up, then suddenly we're worried about it. So in a sense, the intelligence is one aspect, but also the physical capabilities of what the robots can do are another. And that, my worries become more in the military domain. So if we're communicating brain to brain in a way, great, lots of commercial opportunities, but then you put it in the military perspective and it can have distinct effects on humans, it's a different question for me.
Well, then question to everybody is the qualitative difference in magnitude. I mean, we know that when you have huge differences in scale and in the governing systems and the logic changes. We know that from quantum physics, we know that from relativity physics, we know that from many things. I notice that we've had at least sort of three kinds of possible examples in our presentations of different kinds of logic. There was the logic that came from mimicking nature, where we're trying to understand mechanisms, and we go from biology to electronics to robots, and then back to asking questions about nature and so on, and applications. And the logic, therefore, is drawn from that, concluding, for example, that you don't need all these expensive sensors, that you can do something different. There's a logic which says, let us try to build in a way in which predictability becomes possible. For example, the Tokyo subway example of knowing which station you're at. And then there was also the totally inventing a new kind of logic, as Ron mentioned at the beginning, which involved giving robots some sort of ethical sense about feedback loops or that sort of thing that might not just do a logic of implementing a mission, a la Terminator, with whom you couldn't bargain and negotiate, but actually to stop and change the mission under certain conditions. Is that a domain where people who are working on these different problems get together to design logic for future robots, biorobots? I mean, those of you who remember sci-fi, the 3 rules of Asimov on robots?
Actually, in my case, and most philosophers, and not all roboticists, have viewed Asimov's laws as a straw man, but not a practical, workable system. But this doesn't require the invention of a new logic. There is a form of logic. It's called deontic logic, which is a logic of prohibitions, obligations, and permissions. And it exists. Horty's form is one particular type as well, too, which actually, in the kind of system I describe, we don't have to resort to that. But because we can use first-order predicate logic, for example, to be able to address some of these issues. So we're not inventing new logic. We're rediscovering existing and more formal logics to be able to move forward with this.
Anybody else would like to add to that? No? Okay, well, panelists, any last words of wisdom for our colleagues before we take our break? No? In that case, let me just say the following to our audience. I think we have seen today, what we have seen today is the present. But that present has also been a window onto the future. And the landscape is vast, and the distances are huge. It's open, it's wide open, and it raises enormous excitement by the fact that there are so many questions. And as we go forward, every step we take not only answers some of these questions, but raises a whole host of new ones. So in the end, it is wonderful for a place like BioVision to be able to say, this is a wonderful time to be alive, and for all these young people who are here, it's a wonderful time to start on your careers as scientists, because every day, every hour, new avenues of research are opening up, new possibilities for humanity are opening up, and the enormous challenges that we see, from feeding 1 billion people who go hungry, to protecting the environment of the world, to ending wars, to improving the condition of our cities, to protecting our planet. All of that will require the best of science and the best of people's minds. And I think what we have seen today is an exciting challenge to go forward and invent the future. Thank you very much. We'll take our break.